The overall goal of this procedure is to enable a quick, efficient, and inexpensive preparation of polyacrylamide gels in a multi-well plate format. This is accomplished by first sandwiching the gel precursor solution between a hydrophobic coated glass plate and an acrylamide adhesive flexible plastic support. The second step is to peel the gel off the glass plate, which has covalently attached to the flexible plastic support upon polymerization, and let it dry. Next, the dry gel and the underlying flexible plastic support are cut into desired shapes and glued plastic side down to the well bottoms of a multi-well plate or any other cell culture vessel. The final step is to coat the multi-well plate-assembled polyacrylamide gels with a cell-adhesive coating such as a monolayer of collagen type 1. Ultimately, microscopy as well as other cell characterization techniques are used to observe the effect of the underlying polyacrylamide substrate, in particular its stiffness, on cell behaviors.
The main advantage of this method over existing methods is that it allows handling of any stiffness and thickness of polyacrylamide gels, as well as their assembly in a multi-well plate format in a low-cost and time-efficient way not afforded by other methods.
Demonstrating the procedure will be Syed, a student in my laboratory.
First, prepare the polyacrylamide gel precursor solution by mixing acrylamide, the crosslinker bisacrylamide, and deionized water. Dissolve sulfosanpa in dimethyl sulfoxane at 50 mg per milliliter. Aliquot 20 microliters of the stock solution into 10 microcentrifuge tubes. Then flash freeze the solutions in liquid nitrogen.